And I think it's an honor and privilege after so many years to be amongst my most respected teachers and colleagues. And I hope my audio is clear. And
yeah, I think so, Prakash. So, yeah.
Yes, okay. Yeah. So what I'll do is I'll just give you a kind of preamble about my background, and then I'll lead into the topic, because the background is a kind of a sketch of my topic too, in some ways. So I'm going to share my screen.
Oh, 1 minute, I'll have to give you the permission to share screen.
Okay.
Sure. Now you can do it. Yeah. Yeah, you can share the screen now.
Okay. So is the screen visible?
Yeah, visible. Yeah.
Is it the whole screen or is it a part of the screen?
The PowerPoint slide and as well as the others are also visible, the subsequent slides. You need
so it's not in the presentation mode.
Yeah, you have to put it into the presentation mode, you know.
Prakash, if you go to display setting, yeah, just change it.
Display setting.
Yeah. Oh, it's not.
It's not showing up.
Yeah. Okay, anyway, just go ahead with it. It's okay. We can
just
see.
hit escape and then see. Or you can just.
That's kind of strange. Usually you should be able to.
I think that's good enough. Just leave it that way.
This is much better.
Yeah, now
yeah.
This is better.
Yeah, you're through. Yes.
This is better. Yeah.
Yes.
Okay. So I'll once again say hello to everyone, and thank you for coming at this time of the day for many people in North America. And I'm glad to be amongst all the respected colleagues and my previous teachers, and it's an honor and privilege. So with that, I'll kind of give you a kind of a biographical sketch of my thesis work and what I did in MCC. So I came to MCC in '83, and with people like Dyeser and then after that Punser, I got really interested in how plants adapt. That was my main question that lingered in my mind. And so when Dyeser suggested, along with our previous Joel sir, that I should study extrafold nectaries. And so that was my first venture into developmental biology, to understand how organs form. But here, extrafold nectaries are forming outside of the context. And that somehow stuck in my head that the plants have that plasticity. And ever since I've been studying developmental plasticity in plants, because that is the question that got deeply rooted in my mind. And for my master's and the MPhil, I did a lot of tissue culture. Again, the question there was, how can these cells reprogram? How can they adapt and change, which is not common for animals? And so if you look at my, I put my publications just to show the different topics I'm going to cover today. One is plant regeneration. The other one is, I did my PhD on flower meristems because I thought flower meristems are determinate, but at the same time, they form complex set of organs. And that's a kind of a genetic mechanism that the plants use to create very complex structures. So I studied that for my PhD. And then I did my postdoc and then further research as a scientist in NRC and back in the university. I studied meristem genes, and then I studied a lot of embryogenesis. Because embryogenesis is the formative stage when you go from one cell to many cells, but at the same time with an organizational principle. So the organizational principle is driving morphogenesis, and it's integrating certain parts together along with cell division to form a single unit of expression of plant, which we call the embryo. And so the complexity during that process of assembling the different meristems and the ability of the plant to be able to recapitulate that under different conditions, which I call reprogramming, is something that has been at the heart of my research ever since. And so I've been trying to integrate how plants create integrated form and at the same time use the same genetic mechanisms to adapt and recapitulate those programs, because that's a huge developmental plasticity that you don't see in animals. And so the root of the developmental programs, there is a kind of a very modular framework which the plants use. So my journey has been to kind of uncover that modular program. So I did a lot of developmental studies. I studied a lot of mutants, and then from mutants on to gene expression, and then a lot of genomics, because we're looking at seed transcriptome. So we did a lot of genomics, and then from there went to gene regulatory networks. So gene regulatory networks are important higher level logic in the genome, because they are a set of instructions that is used with a plant to either develop or to respond to the environment and develop. So gene regulatory networks are an important part of that. And arising from gene regulatory networks is the concept of what we call developmental modules. So now you can think of each part of the development as a module. And this leads into my topic, which is what I call making, maintaining, and mastering. Because in the embryonic stage, the plant basically is trying to make these meristems and put them together to create one body plan. Then during morphogenesis, in the vegetative and the post-embryonic stages, it is maintaining those meristems and able to produce organs like leaves and flowers and eventually seeds. And independent of that, they also master the act of producing meristems by putting them in axillary places, by producing lateral roots, by producing adventitious roots. Each time they do that, they're building a module of a meristematic module, which can elaborate the architecture and adapt to its environment. So that's why I use that to bring the concept of development and adaptation together. And then obviously we know that plants are extremely regenerative, and that's one of the reasons why we can regenerate all kinds of modules from the plants, either shoot, root, or the whole embryo, or callus for that matter. So this is what I call a developmental plasticity. And so within that, so I want you to remember the word Devo, because it's a conjunctive word associated with evolution. So one of the ways you can study these modules is how they arose during evolution, because the genome, a lot of the structures in the genome, starting from early plants to now, was conserved. So by looking at an evolutionary perspective, what you understand is that these modules were assembled in piecemeal manner during development and during evolution to get various kinds of body plans, starting from the earliest organisms. So this is just a comparison between plants and animals, just to show how the plants are much more plastic in the developmental program. So if you compare plants and animals, plants on the top and the animals on the bottom, you can see that the ontogeny is a sequence of events that leads to the formation of a structure. So during the ontogenic development, there's a dramatic difference, because the embryogenesis is a very short phase in the animals, in the plants. And at the same time, you see only a rudimentary structure of the development happen. And a lot of the development is post-embryonic. But in the animals, if you look, in embryogenesis, most of the body plan is put together. And then what you have is resident stem cells in the organs, which will allow homeostasis of that organ either to grow or to maintain till their lifespan. So that's a fundamental split between how plants build their body and how the animal builds their body. So that's a split. So this is the extent of developmental plasticity you see in plants, because they build modules over time, and that allows the plant to adapt to its environment. Whereas the animals would develop other things like limbs and sensory organs to move and adapt to their habitat. So if you look at the core of an embryonic structure, you can recap that program here, starting with fertilization, with a pollen tube entering the ovule that's inside the gynecium. And then once you have the fertilized zygote, they undergo these kind of pre-programmed pattern of divisions, very conserved patterns of divisions. And this is what we call onograd type. And so the canola, this is an example of canola. And you see that the patterning occurs to give rise to an embryonic structure and an extra-embryonic suspensor. And then the embryo eventually forms these meristematic cells in the middle here, which is the shoot apical meristem here. And then they form the root apical meristem here. And then they link it with this rudimentary unit of a body plan. And so I've just highlighted how the seed is formed, basically. But the core of the topic here is not seed, but just the embryo itself, because what we're going to see is that during the making of these meristems, the plants use a very intelligent algorithm. So if you think of module as a program, and that algorithm is very intelligently used with a plant to be able to put together a body plan, but also maintain the plasticity. That's the core logic of how what I've understood so far about how their adaptive force during evolution has evolved over time. So if you look at meristem, the concept of a meristem, so if you take a shoot apical meristem or a root apical meristem here, so the core of the meristem is called a niche. A niche is a protected environment where the cells are maintained in a particular framework, which allows them to keep producing more cells. So a meristem is a stem cell niche, which has a proliferating zone and a quiet zone in the middle. And then there's always a communication with the differentiated zone. And by having this cross-linked communication, the stem cell zones are maintained throughout development for years together to produce repeatedly organs or to grow as a root. So in the embryonic body plan, you have another structure which is called the procambium early on, but later on, during post-embryonic development, they become the vascular cambium, and they contribute to secondary growth. So you have these three growth centers you can think of that the plants use to build all kinds of modules. So this is what I call maintaining the meristems. Maintaining the meristem is, as they do the job of producing leaf primordia over time, these meristems have a particular developmental module, a genetic module, that allows them to interact with the environment to produce leaf at a particular time based on its stage of development. So much slower early on, much rapid later on, and then completely evoke another program to go into flowering later. So same group of cells is responding to developmental triggers and also to the environmental signals that are coming in to channel its rate of growth. So you have to think of it as a very highly receptive structure. And the same is true for the root meristem. So if you take a root meristem, you have a root cap and you have a hidden group of stem cells here that are responding to environmental triggers and able to sustain growth over a long period of time. So now comes the concept of evolution. The concept of evolution is very important because these genetic modules that assemble these meristems evolved from ancestral plants when the algae was trying to move out of water to land. That's about close to 480 million years ago. They were trying to move out of water to land and adapt to land environment, so it's completely dry. So they have to develop structures which allow them to anchor, which allow them to absorb nutrients, and then grow upwards in competition to get the sunlight. So these were the primary drivers that led to land plant evolution. And so here, if you look at the sketch, this paper is from a 2017 paper where they sequenced the Markentia genome, the bryophyte. And what they find is that a lot of the transcription factors that work in Arabidopsis are already present in Markentia. So the footprints to create what we see as big trees in the current landscape already existed in Markentia in rudimentary form. And over time, during evolution, many of those genes were duplicated and you had a family of genes. So if you look at a particular cluster of genes, then you'll find that a few set of those genes are present in Martensia, and over time they got elaborated, either to build a shoot or a root or whatever, or build a leaf, or many other examples you can think of. So this cartoon is a very important part of our talk here, because it shows how sequential assembly can be visualized. So if you take apical meristems here, the first attempt to form apical meristem was in mosses, in Tiscometrella. It's a model system of a moss, which is a bryophyte. And even in that structure, you have a very rudimentary meristem-like program that can form not true leaves, but leaf-like structures. And then later on, in these structures, there was no vascular tissue. So the second growth center, which can form vascular tissue, evolved later. And the first instance of it they found was in a fossil plant called Algophyron. And later they find that a lot of the lycophytes, what we call Selaginella and Lycopodium, that's where you see the real expansion of the vascular program, and then eventually roots. So the root came as a piecemeal approach, adapting to produce a structure that can anchor the shoot and absorb nutrients. So prior to that, they were only rhizoids. There was no roots. So you can see, so if you imagine now how an embryonic structure would have built. So the embryo has these three modules. You have the shoot, vascular, and root module. So early in evolution, you had a shoot-like module. And then the vascular module was added to it. And then the root module was further reprogrammed from that. So the root actually is a reprogrammed structure of a shoot, if you put the Evo-Devo logic together. So that's the essence of the talk. Will be an Evo-Devo exercise. But what you'll see is that we are really stymied, or we don't have much data. That's one of the biggest challenges. All our data comes from one model system to study genetics. And that's Arabidopsis. Only now people are thinking about other monocot systems as models. So that's a huge gap we have to fill in plant biology. In the animals, we have so many model systems. When you come to plants, all you have is Arabidopsis and a few other plants which we have sequenced, mainly for the purpose of our gain from them, which is what we can get from them, not really to understand them. And that's a challenge that the scientific community will have to overcome in the next decade or so, because we don't really understand the developmental programs, except for one particular model which we have studied, Arabidopsis. And you'll see in my talk also, I'll be kind of stymied because I'll be using only Arabidopsis to explain my model. But essentially what we have to remember is that during evolution, these modules were built sequentially, or at a particular time during evolution, that a couple of modules came together. And then eventually you got an embryonic body plan, which we think of as that we see in the seeds of seed plants. So to keep that perspective, and there's one other example that is important here, which is looking at a morphogen, like auxin. So one of the studies that we have always looked at is understanding of hormones who work in plants. So auxin is a well-known morphogen, a hormone that has been studied to a great extent in the last decade or last few decades. And now more and more genetics has highlighted the role of auxin movement and the concentration of auxin in reprogramming cells. And that's another important part of this particular talk. So this is the Evo aspect of the 4 M's. So in the Evo aspect, you have these 4 different aspects or 3 different aspects of these meristems that are integrated to form the embryo, and in the Devo of how they function. So that leads to the field of Evo-Devo, which allows us to understand developmental modules out of the framework of a single organism, but rather as a family of organisms. So this is a question always that is posed at the interface of evolution and development. So if you look at, so the Evo-Devo question we're asking is, how did a particular development program originate? How did that program evolve to adapt? to become more and more sophisticated or
Yeah.
more and more apt to the particular growth that they are undergoing. And how can that process be modified during evolution? Because that will take us into the future, because once we understand how the processes are modified, we can tailor the crops or the plants better. So it's at the heart of crop improvement. But we completely miss this part because we don't have the tools. Right now, all our tools are genetic tools, so we sequence genes and we study them. But if you know these modules, and if you know how the modules are adapting and growing, then you can use that to potentially improve crops in the future.
இத க்ளோஸ் பண்ணிட்டு தான் அத பார்க்கவா? மியூட்லாம் இருக்கு இல்லையோ?
So in the Evo-Devo field, it originated in the animal system, but there are some key concepts we can borrow from the animal model to co-opt for plant Evo-Devo. And so Evo-Devo happens in the context of an environment, and so
எஸ்கேப்
So there are 4 key terms in Evo-Devo which will be very useful to remember. The first one is called dissociability. So if you take a program like Embryogenesis, it's an ontogenic sequence going from a single cell to a mature embryo. But if you can dissociate modules, then you can start to study genetic modules that are associated with particular developmental modules. And that leads us to the term of modularity. So the shoot is a module, and then the root is a module, and the vascular is a module, for example. And then that's how the form is built. So the morphogenesis that happens during ontogenic sequence is built as modules of set of information in the background, in the genome, coming from the genome and the genes. And as they manifest, you see a continuous sequence of events. But if you're a person who's studying the genetics and the physiology of the organism, you can go back and ask, what are the modules that are driving this formation of this form?
எதை மீட் பண்ணீங்க? இத மீட் பண்ணிட்டீங்களா?
And once you can grasp the concept of modules, then you can make that leap to evolvability. Because the evolvability concept comes from change in the modules or the linkages. So if you have four modules, for example, you can think of each module being tinkered over time, and the linkages between these modules being tinkered to give rise to what we call lasting phenotypic change, which we call adaptation. So the adaptation in the long-term evolutionary logic happens because the ontogeny is a series of modules that can be tinkered with over time during evolution and be retooled for a new environment, a new habitat. And so therefore you have a speciation and adaptation. So that's the core of the Evo-Devo logic. And here, this is a classic example I used to teach Evo-Devo, because if you look at a limb of from bat, whale, horse, frog, take any animal, and the limbs have the same signature formula. It's called 1 bone, 2 bone, many bones, and digits. So 1 bone, 2 bone, many bones, and digits. And you can see how it is being modified during evolution to create various structures from bat to whale to horse to frog to humans. So you can think of how tinkering happens using that as a model. And this has been a very powerful model to understand the evolution of, again, movement of animals from water to land. And one of the fossils, which is called Tiktaalik, was found in the Canadian Arctic. And it was a very popular find in the year 2006. And there's a documentary made on it. And that again highlights on the role of modules in developmental programs and the genetics behind that driving these modules and their adaptation. That's the core of the Evo-Devo. So we'll switch back to plants. And so in plants, we know that we are looking at land plants. So in the land plants, again, we call them embryophytes because they all form embryos, right? Starting from liverworts, bryophytes to the angiosperms. But the embryos over time, in the earliest form, they only form a sporangium, right? In Merkentia, the sporophyte is just a sporangium and a sporangiophore. Whereas once you come to Selaginella, you get a sporophyte, and you have these alternation of generations between a gametophyte and a sporophyte. And eventually, once you end up in angiosperms or even gymnosperms, the gametophytic phase is extremely short. And the sporophyte is what you see as a plant. And so you can keep that in mind. So now I'm going to go into a kind of a dissection of what we talked about in Evo-Devo, using Arabidopsis as a genetic model. Okay. So we talked about an ontogenic sequence, and then we talked about modules, right? And we talked about how you can dissociate an ontogenic sequence in a module. So here in this exercise, should we take Arabidopsis? This is a fertilized zygote, or a fertilized egg that has become a zygote. And here is the mature embryo. So if you divide this, you can divide this into 4 modules as a test case. I'm going to divide this into 4 modules just to explain to you how that will help us understand embryogenesis better than looking at it as a single sequence. So one is what we call embryonic, extraembryonic. Right? Extraembryonic is the suspensor, and the embryonic is the mass of cells on the top, which actually becomes the embryo. Okay. The next module is what we call a shoot and a root module. So if you take this massive ball of cells, which we call the globular embryo, over time, at some point, you find that the uppermost cell of the suspensor will undergo an asymmetric division to form a cell called the hypophyseal cell. And that is the founder cell for the root group. Okay, so the shoot is essentially derived from this ball of cells, but the lower part of this ball, along with the suspensor cell, is contributing to the root group. So you have a second module here where you can dissociate it from this module. Okay. The third module is what I call a bilaterally symmetrical and a radial symmetrical module. Right? So from a ball, you transition into a cotyledonary structure and a hypocotyl-like structure. So you get patterning, and the patterning also involves formation of a unit called a phytomer, a single unit of a shoot-like structure. And the last unit is called the hypocotyl and the root module, because after the phytomer is formed, you have an hypocotyl. So at the junction of the hypocotyl and the root, there is a hypocotyl and root module. And the reason why you have to look at it that way is so now you have dissociated into 4 modules. So you can think of each one of these modules controlled by certain genetic programs. And how can you dissect that? You can dissect that in many ways. You can do experimental manipulation. You can use mutants. You can use gene expression. You can use conditional knockouts. So these are the examples I'm going to show you based on this model. So these are the 2 hallmarks. One hallmark is you have this kind of iterative program where you can form a phytomer, and the phytomer can be repeatedly formed. The second hallmark is that, we know from plants, is that they're extremely plastic. So if you remove a module, they'll replace that module. So if you have a shoot-like structure, you can induce them to form roots, right? And from a shoot, you can form another shoot, right? From a root, you can form a shoot. So these are the examples listed in G. So that is the plasticity. So once you dissociate it from its architectural framework, they can recap and form whatever is missing. So if you cut a shoot, they'll form a root. If you cut a shoot, you can again regenerate a shoot like a bulbil, or you can form shoots from a root. So you have these 2 different hallmarks. One, a very integrated form, and the other one, once you dissociate, reintegrating another module back in. Okay, these are the 2 modules. So essentially in the exercise that I'm going to go through, the reason I chose this as an exercise is because it covers a broad range of topics, but it also informs on the core concept of how development takes place in plants. So one is the modular nature, one highlighted in white. It's a modular program, so it's always forming repeated units, either as shoots or branching roots. But at the same time, it has a very high regenerative potential. And essentially, these two are looked at as two different separate things in plant development. But if you put them back into an dissociable ontogenic framework of embryogenesis, you'll see that these two are actually two hallmarks working together as a single genetic framework. So I'm going to give you some classical examples of genetics and manipulation, just to explain how that can be visualized. So we looked at the two hallmarks. The first hallmark is the iterative program, which is an ontogenic sequence. So we talked about embryonic and extra-embryonic. So the corresponding regenerative or reprogramming potential is the ability of reprogramming of a previous form. The zygote can be reprogrammed to initiate an embryonic program. So I call it activation of the embryonic program. But at the same time, in the lower part of that extra-embryonic structure, it is able to repress the embryonic state.
அதுதான் நான் நினைச்சேன் நம்ம ஏதாவது பேசினா இதுவாயிடும்னு நான் அதை அமுக்காம அப்படியே வச்சிருந்தேன்.
டிவைஸ் மூலமா வேணா ஏதாவது பேசுங்க.
Hello, can you please mute the phone if you can? Thank you.
You mute? 1 minute, I'll just check. Somebody else is. I think everybody should mute their phones. If I mute means even Benglet will get muted, you know, that's why I'm not doing that. Everybody is now muted, you know, so we could go ahead.
Okay.
Yeah.
Okay. So this is the first module we talked about. So or the first step. So you go from a zygote to an embryonic and extra-embryonic fate. And the extra-embryonic fate is called a suspensor. So when we think about suspensor, we think about it as a differentiated state. But in the background of its genetics, it's not a completely a differentiated state. It can be reprogrammed. So it's actually a repressed embryonic program. And so you can show that through experiments. And one of the simplest experiments people did was a simple ablation study. So if you remove the embryo at a particular stage, so once you go from totipotency to pluripotency, if you remove the embryo, then you can ask the question, what does the suspensor do? And in the next cartoon, you don't have to worry about the abstract. As long as you remember the embryonic and extra-embryonic fate, if you look at the next cartoon, so you have an embryo at a very early stage. This is called an octant. So if you remove the embryo physically, which they were able to do in an early stage, you'll see that, so the arrows that is pointing upwards here is the flow of auxin from the maternal tissue up to the embryo. So as soon as you cut the embryo out, the auxin is going to accumulate in the uppermost suspensor cell. And what you would see is, after a few days, it can recover the embryonic program back in that cell. That cell will recover the embryonic fate. And the embryo itself is lost the coordinative program and just becomes a ball of cells. That's in the early stage. So if you take a mid stage, at the mid stage, what happens is the auxin is being fluxed downwards. But as soon as you remove the embryo, the auxin direction changes because there is still maternal auxin available. So the auxin comes back up again, and you can recover the embryonic program where the suspensor cell is reprogrammed into the embryonic fate. But the detached embryo itself will again recover the embryonic program because it is past a particular competency factor. Whereas in the third one, once it reaches past the heart stage, which is the later stage of embryo, there's no longer an auxin supply from the maternal tissue, so the suspensor cell cannot be reprogrammed. So you can see as an example here that the suspensor fate, what we talk about as a differentiated state, is actually a repressed, in the background of its actual genetic state, it's a repressed embryonic state. And so when you knock out the embryo, it can recover. So if you think of that module, then along with the activation of an embryonic program in one cell, there is a repression of the embryonic program in the other cell. And that repression is coordinated with the morphogen. And how is it coordinated? Basically, these compounds like auxin and cytokinin, they have response machinery in the cell, which is described by its own genetics. So here are two examples of that study here. So there are these transcription factors which are triggered by auxin to cause a signaling. And what you find is that in the embryonic cell versus the suspensor cell, these response machineries are intrinsically different. So when you remove the embryo, what is happening actually is the suspensor cell is being reprogrammed to make the auxin response machinery that is similar to an embryo in the suspensor. And that reprogramming allows the plant to become plastic again and recover a fate that would have been normally lost. And through further genetics, you can go through genetics and ask the question, what exactly are the transcription factors that allows these reprogramming? And when you do that, you'll end up identifying factors like what they call helix to helix transcription factors. So you can go deeper and deeper into the architecture. But essentially, the picture I want to give you is that there is a module, a genetic module that controls a developmental module, and that happens at the first step of embryogenesis. And the essential nature of the module is to activate embryonic fate in one and repress the embryonic fate, allowing the suspensor fate to work in that particular context. But if you change the context, the plant can rewire and reassemble the embryo. So that's the essential nature of the module. And that structure, which is activation of a module and a repression of that module at the other end, happens in all the three steps. So if you look at the next step, if you take the next step, you have the shoot and the root step. So at one end, you're forming the shoot. So at the other end of the shoot, you're repressing the shoot fate, basically. And that's how you get the root fate. So the activation of the root fate is possible because you're repressing the shoot fate. Okay, that's the nature of that particular module. And by understanding that, you'll see that you can explain a lot of plethora of data that come at you in a very simple manner. For example, I'm going to show you a series of slides which give you a genetic example of this particular way of programming a module, which is activating the shoot and repressing the shoot and creating a root. So during normal development, you have what happens is the uppermost suspensor cell undergoes asymmetric division, and you get a hypophyseal cell. And that cell is going to be the founder cell for the root program, along with the signals coming from the shoot. So if you look at the ontogenic sequence, so this is how you get it. So this is a hypophyseal, this is a suspensor cell that undergoes asymmetric division. And that asymmetric division gives rise to the hypophyseal cell. So this cell is what is called the root founder cell. And this is what gives rise to the root eventually. So if you follow that program over time, you'll see that that is at the core of the root meristem. But how does the plant design this as a module? And that's what is really intriguing and interesting, because every time you cut off a shoot, you can get a root. So there's a lot of reprogramming going on. But built within the ontogenic sequence is that instruction. It's not an instruction that is de novo. It's built in its embryonic framework. And that's why plants are so plastic. So I'll give you a few examples. So this we already looked at, which is the flow of auxin. The auxin flows upwards in the early stage, and in the mid stage and later stages, it flows downwards. And then we talked about how embryo identity is created. In this example, I want you to look at a couple of genetic modules here. So you have a ball of cells, and in the top ball, you're going to form a shoot. In the bottom part of this ball, you're going to form a root, right? So if you take two core genetic factors, HDZ3 is shown here as an example, and that is a shoot signal. And PLT is a root signal. Okay. The topless and BDL are coming from the auxin pathway. Okay. The topless is a repressor, and BDL is a cofactor. So auxin acts through this pathway. So when auxin acts through this pathway, topless is removed, and plethora is activated. So you can reprogram the cell to form a root. Till that, it's part of a shoot. But how do you uncover this? You uncover this by using genetic experiments. So remember HD-ZIP3 and think of it as a shoot signal and plethora as a root signal. And this one controlling that, okay? I'm going to show you a few genetic examples which will highlight how these things work, essentially. So you can remember HD-ZIP3 is a shoot and plethora is the root. So here is a very interesting study which shows how these modules interact. So if you take HD-ZIP3, you see plethora is an interactor with HD-ZIP3, and plethora is repressing HD-ZIP3 in the basal domain. So if you remove plethora or any signal that represses HD-ZIP3, it's going to move downwards. And if it does move downwards, it is going to create a shoot fate here, or it is going to repress the root fate. These are the two possibilities. So here is a mutant where they created the Rev10D allele is a dominant gain of function of HD-ZIP. And plethora 1 and 2 are loss of function. So you've lost the root module and you've gained an excess of root module or shoot module. And what happens immediately to that particular genetic combination is that you lose the root program. The root program is kind of ablated in that genetics module. So you can understand that the shoot program, when it's too dominant, can repress the root. Okay. In the next set of the same puzzle, if you keep increasing the HD-ZIP, the shoot signal further and further to the bottom, you can ask what happens. And you can use genetic modules or genetic combinations to explore that. And here is an example where they ablated a particular repressor of the HD-ZIP. So now the HD-ZIP signal is very strong in the root part of the module. So you're completely taking away the repression of the shoot program in the base. And you get a seedling with two shoots. So you can replace the root completely with a shoot-like program. So now you can start to look at how the genetics and development work together by creating simple algorithms. So the algorithm is what we design to probe into that. And the algorithm is that you activate a particular program in one part of the unit and you repress that in the other part of the unit, allowing it to be reprogrammed either to back to that state or to something else. And here the something else is the root. So the root program evolved from a repressed shoot program. And that is seen here. So if you take the other factor I talked about is the topless or the TPL gene. And the TPL is a repressor of the auxin pathway. So if you take off TPL, you can transform the shoot into a root. So here is a topless mutant where both the ends are root-like. So it's the opposite of the HD-ZIP. So if you increase HD-ZIP, you can get two shoots. If you take off topless, then the HD-ZIP repression is removed in the shoot and it's completely ablated and you get auxin going up and creating two roots. So now you understand the state of repression and activation in a module. That's an example of it. And here they show how the transformation of the shoot to root is dependent on that plethora module. So you have a genetic reach into the module, and now you can manipulate where it's expressed and how it is expressed. And how that is expressed is the algorithm, which is activation versus repression, which is controlled by auxin. And a combination of these creates a genetic switch. And the switch in the module allows you to create a shoot or a root. And by keeping it in the right orientation, the plant always makes an embryo of the shoot and a root. But in the context of a post-embryonic phase, when it's iteratively growing and producing structures, it is completely repressing the root. And that's how it's building a shoot, essentially. The innate state of how the plant is building a shoot all the time is it has completely repressed the root program in the shoot, essentially. So here is another combination of experiments which show how the root module itself is an uncoupled module. So we looked at HTSIP. Now let's look at plethora, which is the root signal, and ask what happens if you keep increasing plethora. So if you keep increasing plethora, which is the root signal, over time, you'll see, or in the first case, you remove plethora, for example. As soon as you remove plethora, you can see in the circle here. So these are the plethora expression zones here shown in the top panel here. I don't know if you can see my pointer. The panel on the top shows the expression zone of plethora. So if you knock off plethora function, you essentially can remove the roots. And if you ectopically express plethora in the shoot, you can form a root. So you can see how uncoupled that program is. So what they did was they went further and asked, what happens if you keep driving the plethora up, expression up further and further over a threshold, just like they did for the HD zips. So if you drive it, you see that the meristem size, root meristem size increases over time. And once it crosses the threshold, it can form two root meristems, one behind the other. It can essentially place two different engines, one behind the other. Because each meristem is like an engine. Normally the plant will have only one root meristem. But if you push the concentration of a particular root signal over a particular threshold, like plethora, immediately you get two root structures with statoliths and with the root cap and the whole work. So by understanding how these genetic modules work in the context of an algorithm, now you can think about how you can redesign plants in the future. Once you start understanding these switches, you're no longer constrained by what the plant is doing in its natural environment, but rather allows you the freedom to go into a synthetic platform, start designing plants for the future. This is something that we need a lot more work before we can start thinking about it. But that's where this is going to lead to. The synthetic biology of the day is to make more compounds or things like that. But the future of synthetic biology is to make a plant for your contained environment. Because the future is not going to give us a lot of land. That is something we have to agree for de facto, because after our generations are gone, maybe botanists of the next decade or decades from now will be talking about synthetic biology. Because that's the way we are going to produce our food, I think. Because we can't sustain the way we do agriculture to keep producing food for the population that's growing. So the next module I'm going to show you is the junction between the hypocotyl and the root. So if you take so this is the root meristem here, and that's the hypocotyl. So here I've shown two markers on the panel A and B. The second panel shows you the bottom part of the hypocotyl, just before where the root starts. So you can see it's a genetically distinct region based on the expression of a particular marker. And that's the hypocotyl marker, and that's called the collect mark. Collect is the base of the hypocotyl to where the root is attached. And that's shown in the section here where it's marked with a boundary. So this boundary itself, even though it looks continuous when you germinate a seed, is actually reprogrammable. Because I'll show you a simple experiment they did to show that they can form what is called anchor roots. So this is the main primary root. But if you apply a compound called anchoring, which is a carotene-based compound, you can trigger two other roots in the seedling stage consistently in Arabidopsis. So what that means is that the base of the hypocotyl already has set aside some founder cells to form these anchor roots. And this is going to be very useful to understand how this can be used to explain monocot architecture. Because if you look at a monocot root system, it's very different. We call it fibrous. But essentially, it's not different. It's the same architecture that is derived from this module, but it's improved upon because the end of the hypocaudal and the root is a reprogrammable base. Because according to our algorithm, that base is a repressed module. And so you can see here, so when you apply this compound, which is a diapocarotenoid, you can recover that module and start to make these what we call adventitious or anchor roots. So in addition to one primary root, we can form two other roots consistently in Arabidopsis. And these are simple mock experiments where if you ablate the root meristem, you can trigger that root. So this is an adaptive module. So if you look at a plant, normally they would not, Arabidopsis or canola would not produce these roots, but under certain conditions, they will produce these excess roots, allowing better anchor and better transport. That allows them to adapt better in their environment. And the last module I want to talk about is the phytomer. Phytomer is going from a ball to bilateral to radial symmetry. So to accomplish that, they use the same HT-zip module. So if you look at the HT-zip module here, that HT-zip module plays a much deeper role in patterning that ball of cells into a shoot. So if you look at this module here, it's got another repressor, and that repressor is called Kannadi, and Kannadi was called Kannadi because in Tamil, you know, Kannadi means mirror. So the gene was called Kannadi by John Bowman because he was in Bangalore, and he was talking to some student, and they said, because the mutant of Kannadi has two abaxial sides on the leaf. Instead of an adaxial and abaxial side, if you mutate Kannadi, you will get two abaxial sides. And so he wanted to call this mutant a mirror, and so he got the name Kannadi from there. But Kannadi is a repressor of HT-zip. Okay, Kannadi is a lateral repressor of HT-zip, not a polar repressor, but a lateral repressor. And so what they did in the study was, the Kannadi is a gene family. So what they did was they knocked out 3 different genes belonging to this family and asked what happens to the mutant. Because it obviously affects polarity. So in a single mutant of Kannadi, you will get abaxial both sides. So now they've asked, if you take off 3 Kannadis, what happens to the mutant? And what you would see here is, you can see here. So if you take a triple Kannadi mutant, you start to see more leaf-like structures that are formed below the shoot meristem. So where the hypocaudal is, you will start to see small primordia. So that's the cartilage in the meristem. As soon as you take Kannadi, all the 3 Kannadi factors, HT-zip moves lateral. And that's what is shown in the cleared embryos here. And you'll start to get bumps on the side of the hypocaudal. The cell is being reprogrammed. And essentially, what does the reprogramming lead to? To lead to a phytomeric structure. And so you get extra phytomeric units. You get leaf-like units, and these leaf-like units have trichomes. And so you know that they're not cotyledons. They've moved into a leaf-like program. So essentially, you can recapitulate a bilateral symmetry in an inappropriate zone, and thereby understand how symmetry is created. So Kannadi keeps the HT-zips to the middle part of the embryo, allowing a radial structure and a bilateral cotyledon to form. As soon as you move the HT-zip laterally, you start to get bilateral program in the margins of the hypocaudal, and you start to form loops. So you can think of, again, a repressor and activator module and how that works and how the plants use that. Such a simple algorithm with a few genetic switches to create all these units. Essentially, they create shoot, root, and then repeated units of that. And being able to do that at different places over different time is the developmental plastic nature of the plant. So if you think of it in that manner, then you understand that the program is very simple. And here is an example of another study where they show that this reprogramming has a deeper reach. So what they did was they took an embryo and they showed that you have a hypophyseal program. They looked at the genes expressed, it's called transcriptome. And they looked at the hypophyseal program, and they looked at the quiescent cell program, which is part of the root, and then the columella, which is the root cap. And so we showed that this program differentiates over time during normal embryo development. And what they did was, this is a simple experiment they did. They took the root and they ablated the root meristem. They removed the root meristem. As soon as they remove the root meristem, within 72 hours, they can replace that root meristem. The plant will regenerate the root meristem in vitro. And so the question they asked was, as they go from 0 to 24 to 48 to 72, they asked what kind of genetic programs are evoked at these stages. And what essentially they find is that the first evocation is a hypophyseal program. It's almost recapping an embryonic fate. So the hypophyseal state is first, and then it differentiates the quiescent cell and the columella or the root cap, and then the columella and the root cap are completely separate. And by the time you have the stem cell reconstitution. So you remove a root tip and you give it 72 hours, you can recap it. And the way it recaps it is essentially going back to an embryonic algorithm, essentially activating and repressing genes, creating the pattern. And now if you want to kind of use this module as an example to understand how plants diversify architecture, I've shown this as an example. So this is Arabidopsis. So if you apply anchoring, you get a phenotype which you normally not see, but it's embedded within its framework. So you have these two extra anchor roots and the primary root. And if you want to compare this, you can compare it with monocots. Now monocots don't look so different from a dicot, from Arabidopsis. Essentially, they're the same. They have a primary root, and they have several reprogrammed in the cartilage node and in the scutellar nodes to form these extra roots. So when you germinate wheat, you get 5 roots. The reason you get 5 roots is because it's not very different from Arabidopsis. It has put all those genetic modules together to take advantage of the different habitat it's going to adapt to. So that's why the monocot and dicot are different, not because they have completely different genetic programs. And this is something that we can slowly avoid making mistakes in the future, thinking that these plants are very different programs. No, the programs are very conserved, but how they arrange it, the algorithm they use is ever so slightly changed. So this Arabidopsis module is co-opted in many ways. And if you go into the core of this genetics, you'll see that they are very conserved. So when you look at a rice or a wheat root structure, and we say fibrous root system is so different, and so it's so different from a tap, it is not essentially when you think about how the plant is literally operating with its algorithm. So we think of the plant AI, or the adaptive intelligence, or what we call artificial intelligence in our world. The algorithm they use is essentially tinkered, but the program is essentially conserved. So here is the last set of program that I've been studying in embryogenesis was wheat. And I've been looking at wheat embryogenesis in detail. We completed the transcriptome. And then I moved on to study the roots in wheat. And I've been looking at root structure. And if you look at root, so within a matter of 15 hours, you can see the 3 roots coming out of the base of the embryo. So instead of having those anchor roots come much later in a plant like Arabidopsis, or if you take Fasciolus vulgaris. Fasciolus vulgaris, if you look at germination, after 3 days, you get a whole set of cluster roots coming at that base of the hypocotyl, in addition to the primary root. But in the monocot or poaceae, you get a bunch of roots coming out when they germinate. Most people call it post-embryonic roots, but they're not. They're essentially embryonic, except that the algorithm is designed to move a post-embryonic program into an embryonic framework. And that's how they've taken advantage of the new habitat. And now if you keep this structure in your mind, you can expand this logic into many other plant adaptations. So we looked at these 4 modules. I'm just summarizing here. And so here's the summary slide, okay? So this is how can you understand modules? So if you think of each one of these modules we looked at, and we can call them as steps in an ontogenic sequence or a module. So this is the first module, the second, the third, fourth. So if you look at the first module, they're setting aside an embryonic program first. And then after that, what they've essentially done, the plants have essentially architectured, is to create a shoot program, which we call the phytomer program, and create a root program here, and use that same program to build all kinds of roots. That's the beauty of understanding modules, because essentially what the plant has done is they formed the first root in the embryonic program, and they co-opted that same framework, but with different genetic switches. But the algorithm is the same, a repression and activation switch. And the genetic factors are diversified because the ones that operate in the embryo are no longer operating in the pericycle. But that's how they form the root from the pericycle when you get lateral roots. That's how they form shoot-borne roots, which we call adventitious roots. And later you get nodal roots. If you look at rice and other crops, you get nodal roots. And the nodal roots are essentially coming from that same framework. So essentially all these, what we call the root program or the root structure and the diversification of root system architecture is embedded from a reprogramming exercise. And the reprogramming exercise is essentially used when we try to regenerate. So whenever we are trying to regenerate a plant, when we take a cotyledon or a scutellum or a leaf or a shoot and put that in culture, as soon as you put it in the media, what the plant is trying to do is it's activating one of these algorithms. And each one is a root algorithm. And for starting from a root, it can reprogram that back to a shoot or a callus or an embryo. But essentially, when you push the plant out of its integral form, it immediately says, I have to form a root. Because that's what the plants discovered first in the evolutionary path, to reprogram a shoot to form a root. And so that framework was used by plants to undergo rapid diversification to form all kinds of root-like programs. And it is co-opted for all the plasticity you see, essentially, in a lot of these regenerative steps we look at. So here are the two papers, these are the two classic papers, but there are several papers now. The first paper talks about how regeneration takes place, essentially via a root developmental pathway. But they still haven't made the connection with an embryo, which is what I plan to publish soon. Essentially, that embryonic program is where this initially evolved. So if you look at root regeneration of Arabidopsis, if you put a leaf in a thing, the first thing you get is plethora genes, which is a root gene. And later in 2015, this group, Kalika Prasad, is from IICR Trivandrum. He used to work with Ben Shears, and what he has done is he's looked at several sequence of events during regeneration. And every time you get reprogramming of an organ to a callus, and then eventually get shoot or root, you're essentially triggering the root program, which is very ancient. And that's how the plants remember how to integrate, but also plastically regenerate. It is coupled into one framework. And here are examples. The examples in nature you can see is, if you look at maize, you get essentially a whole bunch of roots coming. They call it these crown roots, seminal roots, all kinds of roots. They're all programs essentially evolved from the primary root program and co-opted as a genetic switch. And so if you take it rice, you get many of these phytomeric units, and phytomeric units are shoot-like units. From each node, you get multiple roots. And it's age dependent. So you can see how developmentally this can be regulated as a switch. Essentially, it discovered a switch, and then it designed several algorithms to manipulate that switch, to allow the plant to adapt, to make roots and anchor and grow and do things. And you can see all the examples in nature. You can look at epiphytes, you can look at orchids, you can look at mangroves. All these are essentially a retooling of that ancient program. And the modification can be very diverse, but if you go to the core of it, it essentially is telling us a very simple story that once they found the shoot, they found a way to reprogram the shoot to form roots in different places. And that led to a lot of adaptive radiation we can see in line. And this is a beautiful example that I always like to share, because This is the forest fire in Australia. And if you look at after a couple of months, you'll see that at the base of this shoot, which is completely burnt out, you'll get new shoots. It's a regeneration. And this resprouting is from a structure called lignotuber. And the lignotuber is essentially bud bank. And the bud bank is found in the base of the cotyledonary node, right? So it's again coming from an embryonic framework. So the plants think miles ahead during the development program of how to adapt. And they keep these genetic reserves to bring up when challenges occur. And that's how they adapt in their habitats. They can bring out these genetic programs because they're very simple programs. But the algorithms they've designed is something which is very deep and which we haven't really tapped into. So essentially, to recap the story, so we have the form being built in an iterative manner, and then you have these regenerative capacities. But essentially, you can tie them together into one program. And the more and more you start to tie them together in one program and look at them as modules, you can start to understand how diversification took place from very core conserved modules. So you have core conserved modules, but they're being ever so slightly tinkered and changed. And thus plants have maintained a lineage, a phylogenetic lineage, which is almost continuous. So if you sequence a Markensia genome, you'll find the same conserved units that form the shoot in Arabidopsis in Markensia. And here is a classic example I like to share, which is Selaginella rhizophores. It's an amazing structure. But if you look at the root tip actually bifurcates. So the earlier pattern of branching was sympodial. So if you look at this root branching, it's sympodial branching. So early on, the meristems did not try to form a lateral program in the root. They were just splitting the meristem. And so you can see this is a classical example. So, but the genetic programs are still conserved. So if you go sequence the genome of Selaginella, you'll find Plethora there. And this is the tree of life that I like to show students when I go teaching. I go to schools to share, that we should always listen to the plants because they are like our ancestors, but they are miles ahead because they've adapted to all these habitats. And if you look at all the mass extinctions the world has faced, they've come back. And if you look at any gap in this evolutionary tree, those are all animals which have gone missing. So that is something that, and I say that there's something that we need to learn from them because they are the master adapters. And the genetic programs tell a story that how they've evolved to adapt to different habitats. But the core of it is that the modules are simple, but they are diversified and their algorithms are very adaptive. So I'll stop here because I think I might have gone way over time.
Good. Well, thank you, Prakash. It's an excellent presentation. And hopefully that we will open it up for questions. I think there are some questions already in the comments, if you can see. Well, let me. So there's a question from Dr. Madhavan, what kind of communication exists in plants, chemical or mechanical? Which is probably, he actually asked that very early in the question.
Some of the, so one of the things that has been understood in, so the mechanosensing field is just evolving in the plant world. But in the animal world, mechanobiology has become a part of most of the organizations. Like if you go to most universities, they have mechanobiology programs. The mechanosensing part of, it's just starting, but people are still very skeptical about it. There are a few people who are working on how plants, kind of sense sound and things like that. But most of the research is still on how hormones are the kind of the main growth factors or main triggers. And because they can reach, so if you think of these, what I call as the core modules, the hormones can reach into these core modules and initiate them or suppress them. And that's why auxin and cytokinin are classical examples of tissue culture. And even for rooting, if you take a shoot and dip it in IBA, it will form roots. And so the way they do it is what is phenomenal, because it's integrated into the body plan. And that's what is becoming more and more interesting, that if they can integrate into the body plan, then the future would be that we can help our own food production by creating synthetic plants which can grow in contained spaces.
Any comments or questions for Prakash?
I have a question. Oh, go ahead.
Sure.
Dr. Daya, you can go ahead. You have to unmute your phone, Dr. Daya.
Can you hear me now?
Yeah,
Yes.
yeah.
Yeah. No question, just wonderment at how beautifully you presented and what a cutting edge of research that you are working, and to know that some of our students, and even more than me, for Professor Jai Singh, who had been teaching earlier and even teaching me, for us to see that some of you are in the cutting edge of research in plant biology. Some of it doesn't even make sense to me, but I know overall what you are trying to say and how plants have this great plasticity and evolutionary ability. Thank you for taking the time and continue your good work. Yeah.
I just want to make a botanical metaphor. These are the seeds you planted. They are sprouting now.
Well, that's generous of you, but we plant all kinds of seeds.
Can I make a comment, sir, here?
Yeah, please go ahead.
That's Suresh. I have now gone back almost around 35, 40 years. That was really brilliant. Like, you just took us through a journey. I really loved it because, I just kind of deviated into a completely different field. But listening to you, it was so lucid, simple, we were able to understand, even though the genetic part of it is all kind of over my head because I'd been kind of out of it for a very long time. But thanks for sharing your experiences and your knowledge. Simply to say, it was brilliant. Thank you.
Thank you.
So there's a question. Yeah, there's a question. This is Satish. Like it's in the comment section. Sorry, I will. All right. So it's from Satish. Is there any relationship with root cap and gravity?
Yes, absolutely.
Absolutely, yeah.
Because they have they have statelets and the root cap themselves. The root caps are produced. The root cap cells are called columella cells, and they are constantly differentiating because they are shed as the root grows. And so, if you look at the root meristem structure at the tip of the root meristem, the outermost layer is a stem cell layer. And that stem cell layer is constantly differentiating columella cells. And once a cell differentiates into columella, they immediately start to accumulate statelets. And the statelets is what is the guiding principle for sensing gravity. And there are a lot of interesting papers. If you just Google root meristem organization and root cap and gravity, there's some phenomenal papers which show how plants constantly are regenerating that part.
I have a question.
Yes.
And you mentioned about anchorine, which is a derived regulatory metabolite required for the anchor root formation in Arabidopsis.
Yeah.
Now, my question is it exclusive to Arabidopsis, or can it also be used for other plants?
See, so what happens is the anchoring comes from a family of carotenoid derivatives.
Yeah.
And they're very closely related to a group of compounds called strigolactones.
Okay.
And strigolactones in turn have the signaling pathway is connected to a compound called catechins.
Okay.
Catechins. And catechins are related to smoke signal.
Okay.
So when you have smoke, the catechin compound level goes up.
Mm-hmm.
So the plants have used smoke to evolve this pathway. Essentially, that's what people think. They think that when there is smoke, the plants use this genetic pathway.
Oh, so then in that case, anchorin is something like a junctional communicator?
Yes, yeah. So basically this compound, what it's doing is, it's reaching into that founder cell program. That is a repressed genetic program.
Mm-hmm.
And activating a root meristem program. So there was no root meristem, but there are those founder cells, and the founder cells can be reprogrammed. And here the case in the compound being a dicarotene, a carotenoid compound, makes it link with the smoke signal indirectly.
Okay.
And smoke is known to trigger this kind of adventitious rooting in some plants. So that would be the connection. Yeah.
That's a pretty interesting point that you have just talked about in strigolactone.
Yeah.
Because one of the things that we had been kind of, earlier when I was working in the natural products area was that it is the strigolactone that is, say, for example, in cotton and other kind of plants like maize and all that, which is actually released into the rhizosphere.
Yeah.
That is picked up as a signal by the parasitic striga to
Yeah.
germinate. Sometimes they say almost around 12 years of dormancy of the striga seeds happens in the soil. And once they plow and then put the seeds of, say, cotton or maize, when they release strigolactone into the atmosphere. So it looks like it is both as an internal signal, chemical signal, as well as looks like interspecific signal for, say, for example, striga to germinate. That's pretty fascinating to listen to that particular aspect. Thank you.
Yeah. So in many ways, they seem to have mastered, when they moved out of water. So if you think of plant as a lineage, when they moved out of water, they mastered wetlands. And then they moved into, dry land. Then they moved into the very arid conditions, which is where you get a lot of grasses. And then somehow they also managed to move on to trees. You have epiphytes. And then they moved back on into water, duckweeds. So duckweeds, or the lemna, are recent species, if you look at the branching. So they've mastered the art of going from water to land, and then dry to dry land, from dry land to trees, and trees to back into water. And they do that all without any brain.
So, Prakash, there is a question from Sweta. Carofish and algae are also used as model organisms to understand various physiological and genetic processes. Can you provide any input on comparison of the two model organisms, carophytes and the Arabidopsis?
Yes,
she's just curious.
Actually, the carophycin genome was sequenced recently. And there's a lot of, when you compare, this is an interesting point. So if you compare such widely separated species, you would expect, a lot of genetic difference. And what they're finding is, yes, there's a lot of genetic difference, but if you look at the core modules, the core modules are conserved. So you'll find examples of, for example, a gene like Mansbox gene, which is involved in flounder, is already present in carrot. If you look at a gene like homeobox genes, which are involved in controlling the stem cell factors, they are already present in carrot. So that once they had genetic elements which allowed them to either build structures or to assemble structures, like groups of cells that can divide and produce structures, like meristems and stem cells, those conserved programs have certain genetic factors. And what you find if you sequence genomes is that the number of units of that genetic factor might have expanded in number. But the factors themselves are pretty much conserved. And the reason why it has to be conserved is because the factors work by binding DNA and activating other genes. So once you have that kind of switch that the plants have evolved, what they do is they don't try to discover a new switch. They just play around with the switches.
Yeah,
So one of the examples I always think of is like if you look at an apartment building, the wiring of the apartment building essentially is all the same. But when you look at it from the outside, somebody is working in the kitchen, you'll have the light in the kitchen. Somebody is in the bedroom, you'll have light in the bedroom. But the architecture of the apartments are exactly the same. In many ways at the genetic level, so that's how one of the things we can glean from that is all cells are the same genetic material, but they become different from each other by turning on different sets of genes. That's development. In evolution, what happens is once they evolve a particular genetic module, and the way the module works by binding other parts of the DNA, activating other genes, because that's how these transcription factors work. The transcription factors essentially are proteins that count DNA and turn on transcription of other genes. So they act like a switch. So once they discover a switch, most organisms prefer to keep the switch, but they wire the switches in different ways. And by wiring, they make different genetic circuits. But the genes themselves are same. And that's why when you sequence genomes from Cara and Arabidopsis, you'll find that these units are conserved. That's the underlying basis of why they're conserved.
Yeah, one question is this, that sometimes when we ask one question, it spews out a number of questions related to it. After your answer and about the strigalactones and stuff like that, strigalactones are also involved in detection of symbiotic, fungi and stuff like that. So is strigalactone or anchorin, for that matter, involved in orchid germinations?
They've used. I might be answering out of turn because I haven't read up on the literature yet.
Okay.
But yeah, orchids are again very different. If you look at some of the orchid seeds, I noticed a question pop up in the chat window. One of the questions was, how is orchids different? Some of the orchid seeds are less than a millimeter in size. And some orchid seeds have only 8 cells in them. They don't have a shoot, they don't have a root, nothing, just 8 cells. But one thing they do have, so they don't have any reserved material to grow. But one thing they do is they partner with an endophyte. And the endophyte allows it to get the nitrogen and the phosphorus it needs to build the DNA. And the sugars. And then it starts to multiply and forms a protocol. So it doesn't form an embryo right away. So the few cells will divide and they form a structure called a protocom. And the protocom will assemble a shoot and later a root. So orchid seeds are actually a regenerative program. They're not like a normal embryo. And so when you take a lot of these orchids, they're very tiny. They're like dust. So they essentially have a few cells, but they have the instruction to make the shoot and the root.
So is that the lactone also, the lactone, serine, lactone, compounds are also involved in quorum sensing too, right?
Yeah.
So anyway, I think did Ragul, it looks like Ravi's student. Did you get the answer that you were looking for on the developmental biology process on orchid?
I don't know. I think Dr. Jai Singh wants to say something.
Okay.
Just to congratulate Dr. Prakash on the
Thank you, sir. Thank you.
wonderful presentation. And you see, as Dr. Dayanandan mentioned, it's all cutting edge research nowadays. But I was really fascinated by what you said about the origin of land plants. You see,
Yes.
I've been reading up a few papers about that, and it is said that, you know, more than Markensia, Anthocyrus
Yes.
is a little more closer.
Yes.
Yeah, that's true.
In various mechanisms
Yeah.
to land plants, and also you know, the alga. What's that? Okay, see, not Frichiella, but the other one,
Coleochaete.
is a little more sort of closer. And I was taken many years back, as Dr. Handan pointed out, you know, in those days, Dr. B.G.L. Swami was doing some experiments. He cut off the shoot and put it on cut off the root tip and then put it on the shoot tip and all that.
Gibberellic acid.
To see as to how changes are taking place. I'm really thankful to Dr. Prakash, and I was enjoying the talk. Thank you very much. And it
Thank
raised
you, sir. It's my privilege
memories
to
back.
talk in front of you all.
Very good.
I still remember vividly your fossil stories. And I still, you know, I find that, you know, that's one of the things that has been really captivating, because if you look at a lot of the missing pieces in the puzzle, that is buried in the fossils. Because we still don't know how we got to lycophytes from bryophytes. And a lot of them are in the Rhynie chert, which we call Cooksonia, which we learned in when I was in MCC. Cooksonia, and then Algophyton, and all these things. They don't have root, but they have these kind of multiple sporangia.
That's right. Yes.
And eventually they say that one of the theories is that is called a sterilization theory, where one of the sporangium became sterile, and that's how we got a shoot. Phenomenal how we can think about the problems when we have fossils back in the picture.
It's very fascinating. Yes, it's very fascinating. I'm still working on my second volume, and so these things are working in my mind also. I don't know if God gives me the time. Someday, I hope I'll bring out the second volume. But your talk has just opened up certain new avenues I have to explore and see what new publications are. Thank you very much for that.
Thank you.
Like to, can I?
Yes. Yeah, go ahead, sir. Madhu, sir, go ahead.
I want to congratulate Prakash for the wonderful presentation. And to a broader point that no longer we can study plants separately and animals separately. They have to be an integrated, there has to be an integrated approach to studying these things for a broader understanding of both the biochemistry and molecular biology of these organizations. There is if we teach them separately, then I think we are going to be leaving the students in lurch for them not to be able to connect these things together, especially in terms of cell-to-cell communication and so on and so forth. I don't want to lecture on those things, but I want to say you made an excellent presentation, Prakash. Of course, it is expected of you.
Sorry?
I said it is expected of you anyway, right?
Oh, yes. I hope I lived up to the expectation. It's a tall task, because I remember Madhavan sir and Daya sir's lectures. And, you know, one of the memories that I have still is that when I was a second year student, I was walking down the corridor and I heard this booming, clear voice ringing in the corridors. And then I walked back, and that's when I first heard Daya sir giving a lecture. So, and Madhavan sir's lectures on photosynthesis still vividly alive in the mind. And we talk about it, Pansar and me, we discuss about it whenever we get a chance. Yeah.
Thank you.
Those things are never forgotten. We are always thankful.
It's another comment. I think a lot of questions. Let's see. All right, Janet. Good. I think we were way past the time, but I think it was well spent, I guess.
Yes.
Right.
Yeah, it's really.
So
Yes. Well, thank you very much.
yeah, thank you so much. And then again, I think the webinar organizing committee also put in a lot of work trying
Yes.
to get
I
this
see Leslie
organized.
there. Hello, Leslie.
And Noel for getting the Zoom organized, because it's
Yes,
the day fixed
exactly.
and all that, right?
He's in the pillar to start it. Yeah.
Yeah. Well,
And
we
it's
hope
nice
that
to see everybody in person. It's a wonderful opportunity to see everybody in person and interact.
So yeah, I think we will continue this, and we will get back to you. So I'll get back to Benji, right? So I think we are done with the program right now. I think and then we have some more program in the agenda, right?
Yep.
All right, I'll hand it over to you.
Okay, thank you, Pon. Now, Dr. Joyce, the present HOD of the Department of Botany at MCC, Madras Christian College, is very busy today. He has to visit a few colleges, and so she's unable to be present. So I request Mr. Leslie Lauren to do the vote of thanks.
I think
one more.
I don't know, something on.
Something is wrong.
Yeah, some disturbance is coming. I don't know whose phone it is. Yeah, now it's okay. Oh, still coming.
No, I think it comes from you too.
No, from me.
Only it looks like Dr. Jason wants to say something. Jason, sir, do you want to say something, sir?
He's muted. Sir, you're muted.
You're muted, sir. You'll have to remove the...
Oh, I'm sorry. Yes. No, I just want to know whether this talk will be available as a recording.
Yeah, I recorded it, so there's nothing to worry. I will put it up. I started recording it from the time Dr. Prakash started his message. I should have recorded it right from the beginning, but I forgot. And I just started it when he started his lecture. So, but you will get his talk completely.
Very good.
Included Pon's, adulation, and in the recording, it would have been good, but I somehow missed that. And I just realized it halfway through. So.
Well, what is important is a talk. So
yeah, the talk is
that
completely, yeah.
We got that right. Yeah.
That's very good. So.
Okay. Thank you so much, sir.
I'm sorry.
Fine.
Leslie,
bye. Let me take leave.
Thank you, sir. Thank you very much.
Thank you.
Leslie, the floor is yours.
Sir, okay. I think I think we'll have to drop it, because I think you're not coming through, Leslie.
I think it's probably from Leslie's phone that sound is coming, is it?
Mm-hmm.
Yeah.
Yeah, it looks like that.
Yeah.
Okay. Yeah. I think
it's Leslie's
I'll take the honors.
Yeah.
Thank you, Dr. Prakash, for that wonderful presentation. It took us back to the days when we used to frequent the halls of the botany department. We're so glad and thankful to you. Thank you very much, sir. I'd also like to thank all my the teachers, the former teachers of MCC. Congratulations to each one of you for being able to produce excellent masterpieces like Dr. Prakash and many others. Thank you to each one of you too. I'd also like to thank all the present staff members who are there. And I wish that you carry on, take over the baton from these folks and carry it boldly and strongly. Thank you very much for putting your time and effort to be present here. I'd also like to thank the present students who have all taken the trouble to come, students from different colleges and different institutions. We are so glad that you took the trouble to be present with us. I'm sure you'd be really satisfied and happy with having spent time here. I'd like to thank each and everyone who's worked behind the scenes. Dr. Pon Samuel, when all this planning and things went on, Dr. Nirmala Alexander, I don't know what happened. We were not able to contact her today, and that kind of upset us a little bit. Dr. Janet has been instrumental in planning a lot. I'd like to thank Pastor Noel Prabhuraj for all his effort and all the.
And Dr. Paulin too.
Dr. Paulin, yeah, she did a lot of planning and things. We definitely have to thank her. And apart from that, Vijay, he's been the IT guy for us, helping us to connect with everybody and things. Then Leslie, once again, Mr. Leslie Lawrence for all his effort. And with special thanks to Dr. Joyce Priya, that is Abby's wife, and his two daughters. We also like to thank Dr. Dayanandan and Mrs. Andan for being here and representing Abby, who's not here. It's a pity that I could not catch Abraham, Dr. Abraham. I couldn't catch him over the phone. Otherwise, he would have made it too. Thank you each one. I thank each one for having taken the time. God bless each of you.
Yeah, for Dr. Madhavan also. Dr. Madhavan took time.
Oh, I'm sorry, Dr. Madhavan. Sorry, sir.
No worries. Yeah.
Thank you very much. I mean, it's all you people who were here that produced big people right at the front of the front line of cutting edge technology like Dr. Prakash. We thank each one of you. We'd also like to tell that we plan to have one of these things every month. So if you can,
hopefully, right.
If you can keep in touch, we'd like to. We'll be able to give you more information about it. And those who would like, who are free, I'd like you to add your comments quickly on this thing so that we'd be able to modify if need be and change for the better. Thank you. God bless each one of you.
Thank you so much. Thanks. Thank you. Right.
Thank you.
So everybody goes. Benji.
Yep.
And Leslie, and if Pon can and Pauline can stay behind for a few minutes.
Yeah.
Yep.
To plan for the next thing.
Sure.
Yeah.
Thank you, sir. Sir, you are muted. Sir, you are muted. Madhu, sir, you are muted.
Madhavan, sir, you're muted. We can't hear you. Thank you, sir.
Thank you. I said thank you very much and good night. I'm almost falling asleep here.
Yes, sir.
All right. Thank you.
Thank you, sir.
Thank you, sir.
Thank you very much once again, Dr. Prakash.
Good night.
Thank you, Dr. Prakash.
Oh, it's actually good morning there. Sorry.
I'm sorry.
For us, it's good morning. Yeah, absolutely. It's wee hours, right?
It's good morning here too.
Yes.
Okay. Thank you for the privilege to and the honor.
No, I think it was wonderful, Prakash. I think I think it, it was kicking it off. It's just with good. I think it was actually pretty impressive. I think the engagement was great. So I do thank you for that. And it's a good lesson. I only wish that more folks, especially from the new generation and the students, were there. So it would have been very helpful for them. But I think it will disseminate. So yeah, we took an effort. So thank you very much for that. Thank you, Prakash. Yeah, as always, right? Yeah. All right, Noel.
Yeah.
Then I'll take leave then. Thank you.
Yeah, okay, go
Yeah,
on.
see you. Good night.
Good night.
Good morning.
Good night.
Right.
Good morning. Good morning. Good
All right.
morning.
Yeah, for me it is
1 o'clock?
it's currently marathon. Like I got up at 3 o'clock yesterday, so I heard it's had a busy day today. So
yeah, fun.
Yeah.
If you're feeling tired, it's okay. I just wanted to
I'm okay. I'm okay. So
I wanted to spend some time with Leslie and Benjamin because we are doing the organizing background part.
Yeah.
And
Okay.
if Pauline can also stay back, it will be good. Others are, free.
Yeah, I think Shiva, Manisha, there are some more. Vijay is still there.
Oh, Vijay can be also there with us.
Yeah, Vijay is around.
Yeah,
Vijay is around.
I can't see Leslie.
Leslie is gone. Can you just call him to ask him to come back online?
Yeah, I think we'll see because Leslie's phone somehow it's screwed up.
Oh.
Let's see, he'll come back. Let me see. Vijay is Hey, Vijay.
Vijay, just unmute your phone, my dear.
Right.
Okay. Good morning.
I thought you went to sleep, Vijay.
No, I'm I logged in, but I was not here. I couldn't. I was on and off, and I was in discussion with my team. And
your team, okay.
Not my team, my MD. We have some little problem in the office. We're trying to solve it.
Oh, it's to see which office does have a problem. Tell me. Right, so I want to go there.
There are 2 projects.
I have the same answer, go to an office where there is no problems.
No problem.
I have 2 offices, so I'm now in the office where there's no problem for me.
No.
Less problem.
Yeah, we got to get with Leslie. Let me get my phone and see.
Yeah. Just Benji, can you call Leslie, please?
My phone is somebody called me, so my daughter.
I'll buzz him.
Yeah, I tried calling him. He didn't pick it up. So.
So Prakash.
Prakash is not here. He's gone.
Oh, he's not there.
Prakash left the thing.
Yeah, Prakash probably have to go to work tomorrow morning. So it is totally.
Yeah, I called because we've been planning to meet and discuss about the future thing, future webinars. So we can actually discuss right now about how we are planning to go about it and when do we plan to do about it. And we need to get one person as an MC. Last moment, if you tell someone to become MC, it's a bit of a problem. Because when people were asking me, first of all, I did not know that he had sent that Leslie had sent those prayers, this thing, because I am really overloaded. Yesterday, I finished one last class on graphic design, on 3D modeling. After that, I was completely out. Today morning, when I woke up, suddenly Benji calls me and says, okay, this prayer has to be added. So I added. But what I want is, if you can just do it one day in advance, it'll be easy.
True, very true. It should have been done a little.
And then the most interesting thing is this: at 9:30, Benji calls me to add those prayers in the PowerPoint, and he is also giving me the philosophy behind putting it in. Now I don't have time; half an hour I have to do it, and he's going into the details of why it is being done and etc. That's not needed. And so you just tell me what to do, and I'll do it. Don't give me anything else. Need to say anything else,
I don't know what happened to Dr. Nirmala.
Okay.
I couldn't contact her over the phone. It says not reachable in Varde.
Okay.
So I couldn't speak to her.
Okay.
நிறைய பேருக்கு yesterday too I could not contact many people. Abi இருக்கான் இல்லையா அவன் classmate.
Okay.
அவனை I couldn't contact at all. I tried many times yesterday and even today. அவன் எடுக்க போகவே மாட்டேங்குது. போகுதா he's not answering.
Okay. Now, one more thing is the person who is the MC, you doesn't necessarily have to be the most elderly person. It can be anybody. They just are the persons who will announce who's going to do what. That's all.
Yeah, I think this is actually, you're right. I think this would actually give us the opportunity to just brush up, right? And then to
the previous time what happened was Benji was MCing and then all of a sudden said, Noel, turn ta va ipo. No solra. You have to announce that, man. You don't tell, turn vardu. That's not proper, You just say, okay, now Noel Provraj or Pastor Noel will be, giving us the something. Or now Prakash, Dr. Prakash, like Pon did today,
Okay.
Something like that you have to do. If you say, hey, who's turn next? Noel, turn ta next. No, that becomes that's not in a forum like this. It's not proper. Now
that's FCC style.
So
informal, informal.
Just the announcements. Okay, who's doing what and etc.
I think what we can do. Go ahead.
Don't have to necessarily have Mrs. Nirmala because she is the senior most. We can give her a platform, but let somebody else just do the emceeing, announcing who's doing what, so that there is a flow to the program. And the MC is also the moderator. Yarad konja jasti poitanga, okay, now let's take a break and we'll hand it over to so and so and so. Andamari, they are also the moderators,
So we'll have to also have backup.
So always have two people. Okay?
Yeah, two people.
So one person is the main person, and if they are not, then the next person will handle it. And both of them should be in the complete loop about what the program schedule is all about. Because today morning, I don't know what the program schedule is. I know that Dr. Prakash is speaking, but besides that, what is coming before and what is coming after, I don't know.
Yeah. So what we can do next time is also a little, I think.
Leslie, sir, come into the Zoom.
Actually, talk to Margaret. See, Margaret is not a person that
come into the Zoom. Sir is calling you. Noel sir and
like go to bed at like 1 o'clock.
You're
right?
in?
So she won't do it. So we have to like figure out a time. What is in for her? And she said 1 hour, maybe like 9 o'clock. She said like 9 is 10:30. Like okay, she is actually in the East Coast time, which is so today this time we started at 10:30. It would have been 11:30 for her. For me and Prakash, see, Prakash is right above north above me, right? Saskatchewan, the same time zone. So it was, but see, that's again, it's just it's a big wrap, right? So, I mean, I don't want her, like you guys, she's good, but I think she's not like,